Next, I would like to invite Dr. Kymora. Dr. Kymora Scotland is assistant professor at the University of California in Los Angeles. She's an endourologist focusing on kidney stones and BPH. Thank you.
Thank you. Okay, so this will be a little bit more of a basic science talk, but I'm going to try to make it as applicable for the audience. So, this is some of the work that I do in my lab. I'm also a urologist, but I do some work looking at how kidney stones form. Okay. And these are my disclosures. I should mention also that a lot of the work that I do is as a result of the work that I did as a postdoctoral trainee. And my postdoctoral, some of my work was funded by Dr. Raju Thomas. So always want to thank him for that. Okay, so I'm very interested in how kidney stones form. Kidney stones affect about 10% of the global population. And despite the fact that we are so used to kidney stones, we really don't know what causes them. Um, we have a sense that there are many things that contribute to stones forming. So, diet, as urologists and as physicians, we talk to our patients all the time about what they should eat and not eat to prevent kidney stones. But it turns out that it's not just about diet, there are genetics involved, hormones, and potentially bacteria. So, we do know that successful prevention of kidney stone disease is going to require us to have better targets for prevention, because right now what we do for prevention is not very helpful. The most prevalent, the most common stones are calcium oxalate stones. And so the question is, you know, how do those stones form? Now, we know that there is a connection between infections, urinary tract infections in particular, and kidney stones. So, kidney stone formers are at a higher risk of UTIs. And then, when we have patients who have kidney stones and have recurrent urinary tract infections, when we remove those stones, their infections decrease. So we know that there's a connection. But typically, what happens for urologists when we see these patients is the following. So we will have a patient who comes in with a stone that is obstructing their kidney and they have an infection. Or we have a patient with a very large stone that we think has been there for years and got infected. And so maybe that's why that patient has infections. But then you also have patients who have things like struvite stones, which are a very rare form of kidney stones, which we know are formed by bacteria. They include bacteria and they are infected. But like I mentioned earlier, 80% of stones are calcium oxalate stones, which we do not think are infected and only see infections in those patients, we think, when there's an obstruction. So I am here to argue that that is not actually true and that there are data to support a role for bacteria in calcium oxalate stones, even in patients that do not have infections. And so this goes back to a few years prior when the urobiome was identified. So you have all heard of the microbiome, and several years ago it was found that bacteria exist within the urine. Previously, urine was thought to be completely sterile. We now know that that is not true. And so we started looking at kidney stones, even in patients who were not infected. And we found that those stones, if we did next generation sequencing, we would identify some bacterial populations in those stones. And so then that led me to think that potentially those bacteria are there for a reason, and that uropathogens, so the bacteria that actually cause infection, might be promoting these calcium stones to grow, and that they might do that via biofilm formation. I'll explain in a little bit what that is. So we had three objectives for this talk. One is to first, well, if I think that's true, identify these pathogens, and then to figure out how they might be playing a role. So that takes me back to geology. And I should have mentioned previously that I also have an appointment at the Natural History Museum in Los Angeles. And the reason for that is because I'm very interested as well in how geology works with bacteria, because we have stones that we see in places like Yellowstone, where those stones were grown as the result of bacteria. Bacteria deposits minerals, and those minerals layer to form stones. And so my question was, well, why would it be any different in humans? Right? Nature replicates things all the time. Kidney stones have very similar layering to what you see here. And so what we did was to first use a fusion of X-ray microscopy, so various techniques to image the stones to identify whether they are really, in fact, similar to what happens in geology. In geology, what happens is this process called biomineralization. So, biofilm is this deposition that bacteria will make. So, they deposit these materials and they cause minerals to then deposit along the biofilm, and then they form these layers. So, we hypothesize that this is the same thing that's going on in kidney stones. And so, what we did was to look at the substructure of the stones. And we used here this mixture of X-ray microscopy and X-ray fluorescence and micro CT, which are all these imaging techniques. And what we see here is something kind of similar to what I showed you in the last picture, where you have all of these layers. So for instance, sorry, let's see if I can go back. So for instance, here you start to see this central area, but it's surrounded by different layers, and you see it again here and you see it again here. I should mention that down in the right is a struvite stone. So struvite stones, as I mentioned earlier, are the stones that we know have bacteria. But these stones in the top and to the bottom left, these are calcium oxalate stones. So they look fairly similar. And you start to see these areas, different areas here, where maybe you have, I don't know if you can see it down here, you have this area that's surrounded by layers, another area surrounded by layers, and on and on and on. Starting to think that, well, maybe there's something causing the layering in each of these areas. And maybe that something is bacteria. So what we did was several techniques to prepare kidney stones that I got from my patients, and we broke up the kidney stones. And what you're seeing here is again advanced microscopy. So this is scanning electron microscopy, which is a way of really looking at really high resolution at these stone fragments, and we were able to identify bacteria. So what I wanted to show here, I mentioned earlier that my hypothesis is that even in patients who do not have infections, we're going to see bacteria. So what I want to mention here is that on the right lower quadrant, that comes from a stone, okay, where there was actually an infection. Now, fragments left, left top, left bottom, and left right top, these came from stones where there were no infections. The green that you're seeing here are actually bacteria. So we're seeing cocci, and we're seeing also biofilm that I mentioned here. So in the stones, not on the surface of the stone, within the stones, we start to see bacteria. And so this is something that really hadn't been shown before. But nobody would believe us if I just showed that. If I just showed this and said, oh, they look like bacteria. And so what we needed to do was to then stain for elements of biofilm and elements of bacteria. So bacteria, like every other organism, have DNA. And so we stained specifically for bacterial DNA. And so what we're seeing here is this. So in this top corner, what you're seeing is just the piece of stone without really any staining. On the bottom two here, I'll show over here, maybe I'll try. On the bottom two here, this is coming from a stone that we got. Remember, I said that I work at the Natural History Museum as well. We got this stone from a quarry in Germany. That's a calcium oxalate stone that did not come from a human. Calcium oxalate also exists in the world. And so you can see here that when we stain it, we don't see any bacteria. which makes sense. This is abiotic. But then in the kidney stone that we got from patients, we start to see staining, right? So the green here is DNA staining for DNA bacteria. And what we're seeing is that it's not just bacteria in the stone, but the bacteria is layering, kind of like the layers that I showed you earlier. And what we're seeing is that there are layers of minerals, so calcium oxalate crystals, and those are the purple layers. But those green layers are organic layers. So it's not just bacteria, but it's also organic material, the biofilm that I mentioned earlier. And we're showing that they're layering together. And so the question is, well, why? Why are they layering like this? And then I wanted to show that it's not just bacteria, it's not just, potentially biofilm. I wanted to really prove that this was biofilm. And so we stained for some of these residues that we see typically in biofilm, one of which is mannose from some of the polysaccharides that you see in biofilm. And this is just to show here that we see that staining as well. And so on the right is really just this square area here magnified. And you see just this mixture of polysaccharides and DNA and all of the things that we expect to see in biofilm. Okay. But what about inside the stone? So I'm showing you the surface of a piece of stone. What happens within the stone? And so we used another type of imaging called FibSEM, where we're able to basically cut through layers of the stone to show you that, again, we do see bacteria. And so the bacteria here is in yellow, and in blue, you see just the crystal, calcium oxalate crystals. So within the stone, you have these areas of bacteria as well. So it's all through the stone. It's not just on the surface. And so that starts to make me think that, well, maybe bacteria is really what's starting these stones growing, because it's all throughout the structure. This is something that we had been thinking about for quite some time, because some of our colleagues had noticed in stones that they see these empty areas. And those empty areas that you can kind of see here, these voids, we've started to think, well, maybe what's happening is that you have stone material, it dissolves, it reforms, and then you form these sorts of layers going around and around because of that. And potentially this dissolution has to do with the fact that the bacteria is sitting there, laying down biofilm. We're working on that now, but that's our hypothesis. Okay, and so, this is just sort of a video to kind of show you those voids. You're seeing the holes now within the stone, which is something that, hadn't really been appreciated previously. Okay, and then the bacteria are not just there. They didn't just, start the stone and then die at some point. We were able to show that they are alive. When we were able to extract bacteria from the stones, we were then able to grow them up, which means that they continue to exist. Now, how they exist in stones without that much oxygen is the question that we're following up on now. But what that means is that these bacteria could potentially be a source of infection, and they could potentially be, an issue for our patients as we treat them. What we do is we break up stones. So we're potentially allowing bacteria out into the urine and potentially causing infections. This is just another way of showing that we have these holes within the stone and starting to think about what that means with the bacteria. So here in this bottom tile on C, what I'm trying to show here is that in the organic areas where the bacteria lives, you have really small crystals forming. But then outside of that organic layer here, the crystals are much larger. So that again can explain how it is that you form these layers and they become these circles that become wider and wider away from the center of the stone. So we're starting to think not just about how the stones form, what the bacteria is doing in the stones. And then the next question is, okay, fine, if you think they're doing something, what are they doing? And so we had to go back a little bit to in vitro work to try to get a sense of how. So we went back and we created calcium oxalate crystals, and then we subjected them to bacteria to try to see, well, what happens to those crystals. And so first of all, what we found is that when you grow these crystals in the presence of bacteria, the number of crystals is increased, the size of crystals is increased, and the aggregation of crystals increases, which makes sense. The way stones form is that crystals aggregate, pile on top of each other, and then the stone forms. So it seems like bacteria expedites the process. And then finally, bacteria changes the shape a little bit of the crystals. All the way to the right is just Raman spectroscopy, just showing that these are in fact the same type of crystals, even if they look very different. We used focused ion beam SCM again to kind of cut through the crystal to see what's going on, because the crystal looked very different. And what struck me is the fact that the center of the crystal here, you can see, almost looks like there's nothing there. You can see the external periphery there. And this is crystals grown in Pseudomonas, crystals grown in E. coli. These are both uropathogens that we see in the urine all the time. But it looks like there's an empty hole in the middle, and we're trying to figure out why. And so we took a closer look, and you can kind of see that here. So here's the calcium oxalate crystal on the periphery, and in the middle, this is just sputter coating that we use to keep the crystal intact. In the middle, you can see there's just this empty space. And so what we think is happening is that the bacteria is sort of, causing deposition of organic material, again, the biofilm, and that pushes the crystal out of the way. And what that does, you'll see in a little bit, is it changes the way that the crystals aggregate. So this is control crystals. They're sitting there, they aggregate by basically, sort of side by side, coming together. When you look at the crystals that are grown in the presence of bacteria, you can see the difference already. They start to grow within each other, which we call twinning. And then when they aggregate, you can see it's a much more complex aggregation. It's bigger piles. And so we think that this is what bacteria does. First, it nucleates the crystals, so it causes them to form in a slightly different way. And then it causes those crystals to grow into each other, so it makes for a much more stable complex, and that complex will aggregate and grow on top of each other. And we think that that is how these stones form. And so what we're doing now is to get a better sense of what this process looks like. But so far, what we think we know from this work is, first, that live bacteria exist within calcium oxalate kidney stones. And those bacteria deposit biofilm within the stones. The bacteria also increase the number of crystals, and they increase aggregation and the complexity of the aggregation of the crystals. So what we're doing now is we're trying to do this in a model that looks like the human kidney. So we have kidney cells that we've put together, and we have artificial urine, which is flowing through like you would expect urine to flow through in the kidney, and we're looking at how crystals form, and we're trying to grow stones in this model. So hopefully we'll have some more data to tell you soon. And those are the questions that we're trying to continue to answer. Why do bacteria do this? What does this mean for the cell? And how does that help our understanding of biomineralization in kidney stones, but also in other organs? So, lots of what happens to our patients with kidney stones is that we don't quite understand why they keep forming them over and over. If we can have a better understanding of how bacteria is working to form these stones, potentially now we have new targets that are not antibiotics that we can then use to prevent stones, and potentially even to treat stones that have already formed. All of this work was done with multiple collaborators, and I particularly want to thank my team members, my postdoc, Rehane, and several of our students and technicians in the lab. Okay, so I'll end here, and I think I have a minute for questions. Okay, thank you. Thank you.